Hello everyone, welcome back to my channel Xgene and proteinX. In this tutorial video, I'll discuss about the history of DNA discovery. So here I would like to discuss 5 different scientist discovery that all are associated with DNA discovery. So first, I want to talk about the Mendel's discovery that associated with the gene discovery. So in the second half of the 19th century, Mendel's work on the inheritance of traits in pea plant suggested that the specific invisible factors are responsible for transferring organismal trait between generations, and those invisible factors were established as genes. So Mendel, he worked on pea plant on 7 different characteristics. Those were plant height, pod shape, color, seed shape and color, flower position and color. And accordingly, what he observed and according to his data, he suggested that the crossbreeding in between yellow pea and the green pea, and he observed that the offspring in between the crossbreeding of yellow pea and green pea always produces yellow seeds. However, he found that the next generation, the green seeds reappeared. Although the number of these green seeds were very lower compared to the yellow seeds, but the ratio of these green seeds, it was 1:3. And this experiment he termed as the recessive and dominant. So as we know these days that recessive and dominant gene, so this is well known. So next is the discovery of the Avery, McLeod and McCarthy's discovery. So those group of scientists, they worked together and they initially assumed that the proteins were the hereditary material. So according to their experimental demonstration, they reported that the DNA is substance that causes bacterial transformation, and they reported in 1944, and according to that report, they widely believed that it was proteins that served the function of carrying genetic information. And this research was conducted at Rockefeller Institute of Medical Research. So next is the Friedrich's discovery. So a century later, Friedrich discovered that DNA is the carrier of genetic information, and he was the first scientist who isolated nucleic acid in 1869. So what next? The Friedrich's findings paved the way for research uncovering the chemical nature of DNA and the rules for encoding genetic information. So for that reason, Watson and Crick, they encouraged to figure out the molecular structure of the DNA. So their model's name, that their discovery that they found on DNA structure, that known as the Watson and Crick's model, and they proposed the double helical structure of DNA. So in 1962, they jointly awarded the Nobel Prize because of their discovery molecular structure of nucleic acids and its significance for information transfer in living material. But the one of the scientist's name, it's not that much well known, and that scientist's name is the Rosalind Franklin. Her discovery and her research, and she has the major contribution on DNA structure discovery. So in subsequent years, it has been recognized that Watson and his colleagues did not properly attribute colleague Rosalind Franklin's for her contribution to the discovery of the double helix structure. And also it was got to know that Watson and Crick, they used some data of Franklin without her consent. So this is why her name is still in dark, and that's what she is known as the dark lady of the DNA. And also she was known as the unknown scientist. So let's have a look at the Watson and Crick's model. So Watson and Crick's model contain 4 features. The number 1 feature, That is, a DNA is double-stranded helix with the two strands connected by hydrogen bond, and A bases are always paired with T, and C bases are always paired with G's. So as you see in this figure, that A always paired with T and it contained two hydrogen bonds, and C always paired with G, it contained three hydrogen bonds. And number 2 feature, the most DNA double helices are right-handed. Only one type of DNA called zDNA is left-handed. Feature 3, the DNA double helix is antiparallel, which means that the 5' end of the one strand is paired with the 3' end of its complementary strand and vice versa. So as you know in this figure that the 5' region always complementary with the 3' region, and they paired with each other. Number 4 feature, that not only are the DNA base pairs connected via hydrogen bonding, but the outer edges of the nitrogen containing bases are exposed and available for potential hydrogen bonding. So this is all about the 4 features of Watson and Crick's model. So also the recent scientists, they made some modification on it. So I'll make another video that what kind of modification they made in recently. So number 4 is the Arthur Kornberg's discovery, and this is the number 5 and last discovery in my discussion here. So subsequently he isolated of DNA polymerase, the enzyme that catalyzes the synthesis of new DNA strand, and he did it in 1956. That DNA polymerizing enzyme that is known as the DNA polymerase 1. And he and his colleagues identified many different components of biological DNA replication machinery. And first he worked on E. coli, and later on he also worked on eukaryotic life forms. So this is all about the history of the DNA discovery. These 5 scientists' names are closely related with this DNA discovery. So thanks. So if you like this video, kindly hit the like button, share it, and subscribe my channel. Thanks.